Thank you so much for the first speaker. And we're going to move on to the second one. The second speaker is Zijian Dong, who unfortunately cannot be here due to visa issue. So he has a recording. I will do the question and answer. So Zijian Dong is a PhD student from National University of Singapore from my group. Please.
Hi everyone, this is Zijian Dong, a PhD candidate from NUS, supervised by Professor Juan Helen Zhou. My talk is titled Brain Dynamics Foundation Model, Pretraining and Adaptation for Disease Prognosis and Treat Prediction. I apologize that I cannot attend in person due to visa issues. I was very much looking forward to being there and engaging with fellow researchers. Although I couldn't join you on site, I've recorded this presentation to share our research with you. A key innovation in the field of AI for neuroscience is the development of brain foundation model. It is a kind of large-scale AI systems trained on extensive brain imaging data that can be adapted for multiple applications. Similar to how language models revolutionized NLP domain, these models learn general repetitions of brain activity that can be fine-tuned for various downstream tasks. In this talk, I'll demonstrate how our brain foundation model can predict individual traits and clinical outcomes from brain data, and how AI models can decode what we see in our brain. First, I'd like to introduce our recently developed brain foundation model, which demonstrates exceptional performance in predicting both individual traits and neurological disorders diagnosis. The model is named BrainJPA. It is a brain dynamics foundation model with gradient positioning and spatial temporal masking. AI is experiencing a paradigm shift from task-specific training to building foundation models that are trained on extensive data using self-supervision at scale. Unlike the models with singular functions, foundation models can be adapted to a diverse array of downstream tasks. In this work, we are interested in brain fMRI signal. We specifically address three critical questions or challenges. How to locate different ROIs in transformer? Does direct reconstruction of mask fMRI input make sense? And how to efficiently mask fMRI input during the pre-training? To address the first challenge of locating ROIs in transformer, we introduced brain gradient positioning, using brain gradient as a functional coordinate system for brain inattention models. We begin with fMRI time series. From this time series, we can construct a functional connectivity. Each row or column represents the connectivity profile of that region. We can calculate the cosine similarity between different connectivity profiles, leading to an affinity matrix that quantifies how similar the overall connectivity profiles are between different brain regions. The functional connectivity gradient is calculated through the diffusion map of this affinity matrix, which is a continuous measure that captures the functional relations among different ROIs, where relative distance between different ROIs indicates the similarity in their connectivity. In the figure shown here, brain cortical regions are situated in the top three gradient axes and colored based on their positions. The brain gradient positioning reflects functional network architecture, such as default mode and visual networks. Here, we show three common self-supervised learning architectures. I will not go through the details of these frameworks in this talk. Instead of reconstructing every detail of the input or relying heavily on data augmentation, we adopt joint embedding predictive architecture, in short JPA, where it predicts the repetitions of various target blocks rather than reconstructing the mask input, enhancing higher level abstraction and boosting scalability and efficiency. As fMRI data has complex patterns across both space and time, we design spatial temporal masking to inject a strong inductive bias during training. After petrifying and shuffling ROIs, we mask the fMRI data into three regions: cross ROI, cross time, and double cross, forcing the model to predict across different brain regions and time points. This structured approach leads to faster convergence and better downstream performance compared to random masking. Now let's see the overall architecture of BrainJPA. During the pre-training, we train the model to predict the target blocks given the observation blocks in the latent space. After petrifying the fMRI data and applying spatial temporal masking, it uses Brink gradient to encode RY locations and predicts target reputations given observation. The model learns through MSE loss, with the target encoder updated via exponential moving average of the observation encoder. BrainJPA was pre-trained on 80% of UK Biobank resting state fMRI data. On internal tasks using UK Biobank's 20% held out fMRI data, BrainJPA significantly outperforms previous approaches, achieving better performance in both age and sex prediction. On external evaluation across different datasets, BrainJPA shows strong generalization capabilities. On HCP aging dataset, we improve significantly in both demographic and trait predictions. Neuroticism measures emotional instability and sensitivity to stress, while the flanker score assesses the person's ability to focus attention and ignore distractions. For clinical applications on ME and MCC datasets, BrainJPA maintains strong performance. Mild cognitive impairment, MCI, is cognitive decline beyond normal aging but without significant functional impairment. Amyloid status indicates the presence or absence of amyloid beta plaques in the brain, a key Alzheimer's disease biomarker. Importantly, despite being trained on Caucasian data, our model generalizes well to Asian cohorts. Now, let me walk you through some further analysis. Firstly, we conducted model scaling. Here, y-axis shows model performance, while x-axis shows model sizes. We observe consistent improvement as model size increases. Secondly, we compare our model with previous brain foundation model, BrainLM, regarding linear probing performance. The blue line is corresponding to our model with pink line for BrainLM. We can see that our model demonstrates smaller performance drops between fine-tuning and linear probing. Thirdly, we perform ablation study to compare three positional embedding approaches: sine cosine functions, anatomical locations, and our brain gradient positioning. We can see that brain gradient positioning outperforms the other two traditional positional embeddings. In the last ablation study, we compare our spatial temporal masking with vanilla random masking regarding performance over different training epochs. Here, orange lines represent our spatial temporal masking, with dashed lines show the vanilla multi-block sampling trained for 300 epochs. Spatial temporal masking demonstrates faster convergence and better performance compared to multi-block sampling. Network level attention patterns in NC or MCI classification highlight critical networks consistently across both Caucasian and Asian cohorts, aligning with known neuroscience findings. To conclude for this work, BrainJPA introduces three key contributions: prediction in latent space, brain gradient positioning, and efficient spatial temporal masking. These contributions lead to significant impacts: state-of-the-art performance, strong cross-ethnic generalization, robust scaling, and improved efficiency with better linear probing. In the next work, we want to answer another question. Given the foundation model, how to efficiently adapt it? We propose scaffold prompt tuning, which is a framework for efficient adaptation of fMRI pre-trained model. Given the pre-trained fMRI model, researchers can fine-tune these frameworks for various downstream tasks. However, there are certain challenges in fine-tuning fMRI pre-trained models. Firstly, fine-tuning could be computationally intensive. Secondly, there is a risk of overfitting during the fine-tuning. And lastly, the previous work has shown that fine-tuning could potentially distort the feature space gained from the pre-training. Prompt tuning represents a promising solution to address the aforementioned challenges. Here is a comparison between model tuning and the soft prompt tuning. In the fine-tuning, it requires adjusting all parameters in pre-trained models and maintaining separate model copies for each downstream tasks. In contrast, soft prompt tuning freezes the pre-trained model, while appending tunable soft prompts to the input sequence, dramatically reducing computational resource requirements. This efficiency makes prompt tuning particularly valuable for deploying foundation models across diverse applications. Our model contains two stages of the training, including source training and target training. In source training, we leveraged high resource tasks with abundant healthy control data. In target training, we applied the knowledge gained from the first stage to low resource tasks like disease diagnosis with limited samples. Here we see how our method works during source training. We start with modular prompts, which serve as fundamental building blocks. These modular prompts are combined based on attention values to create specialized phenotype prompts. The frozen foundation model processes these prompts along with the input data X to generate predictions. The attention values are calculated between modular prompts and input data. The right part shows target training, where we leverage the knowledge in phenotype prompts gained from the first stage. The phenotype prompts are further combined to create vertex prompts that are used for downstream tasks like disease diagnosis. This table shows our experimental results comparing our proposed prompt tuning method in short gaps against existing prompt tuning approaches for two neuroimaging classification tasks, including control and MCI classification and amyloid positive and negative classification in cognitively normal participants. The results show that SCAPS outperforms previous approaches with very limited training data, and the performance scales up with increasing training data. Similar observation can be obtained for the neuroticism score prediction on UK Biobank. We provide some further analysis on the model interpretation. In the left panel, we show that phenotype prompts naturally form into three clusters: personality, social emotion, and cognition, without prior supervision, indicating that they effectively captures different pillars of brain behavior associations. In the right panel, it shows the distribution of attention scores in target training. The model focuses on cognition and age for neurodegenerative disease diagnosis tasks, where it focuses on personality in neuroticism prediction. Looking at our ablation study on the left, removing any type of prompt from our full framework decreases performance. The right plot demonstrates our model's remarkable efficiency. SCAPT could achieve the best performance while using only 1.4 million parameters, compared to traditional fine-tuning which uses 68 million parameters but performs much worse. Lastly, I would like to touch on one major breakthrough in our lab, which has made significant advances in non-invasive mind reading using generative AI. We can actually decode what we see from our brain recordings. These works were done by our recently graduated PhD student Zijian and our collaborator in COHK Jiaxin Qin. Using generative AI, we have demonstrated the possibility to reconstruct image or video from brain recordings like fMRI. Here's an example. A person's brain recording of viewing a giraffe is fed into our model. The benchmark reconstruction is in the middle. Our approach, clearly showing a giraffe, is on the right. We are the first to achieve semantic correctness across all kinds of images, including human, objects, and scenes. We have also extended to video reconstruction, which provides a more naturalistic setting and mirrors our daily life. Here are some example videos. So, what's next for our brain foundation model? In the next phase, we aim to build a multimodal brain foundation model by integrating genetics, text, and other data types. We'll also investigate how the brain evolves over time, capturing key patterns in aging and development. So that's all for my talk. I'd like to acknowledge my supervisor, Professor Juehelen Zhou, for her guidance throughout all of these works, and all the other co-authors. Our research utilizes datasets from UK Biobank, ANI, HCP, and MECC. Finally, I'm grateful to all our lab members for their support. Welcome to visit our lab's website and ask to find more. Thank you for listening. We are happy to take any question.
Any questions? I saw. Oh,
Hi.
okay.
Thank you very much. Very nice presentation. Actually, I have a question regarding BrainJEPA. You use brain gradients as positional encoding. My question is why...
Hi everyone, I'm Xin Hui.
Okay, so why you did compute the positional and the brain gradients only once across subjects, and why not brain gradients for each subject separately?
That's a very good question. Yeah. So right now we didn't do that because there is inter-subject variability, and computationally it's very extensive. So we only try on the group level functional gradient first to see whether it's actually indeed improved the performance. Of course, the next step we could try the individual functional gradients, but it is also be prone to some of the errors at the individual level.
Okay.
Yeah.
Great. Thanks.
I think we should move on. We can answer more questions at the end.